I come from Barcelona. I've been, my studies were on physics. We discussed this morning. But then I prefer to go to molecular biology. You know, molecular biology is founded by physicists like myself. So it is, and then I started to pivot towards more functional biology. And at the end, we decided to go to genomics. My past work was to coordinate the first genomic project ever done in Spain. It was in Melon. I will show a slide on that at some point. But I see my talk today saying the program that you are having these days is more on thinking of things that scientists are asked to do besides their own scientific work. We are asked many times to answer questions that people have. One of the main questions, you know, that we had, especially here in Europe, has been about GMOs, genetically modified organisms. Still, you know, this discussion is hot at this moment in Europe. We will discuss this thing a bit. So, as many of you have been asked to work or to discuss or to give your arguments about many questions regarding food production. And so what is the role of scientists in this? Is this the basis of my talk? I will not talk especially about specific scientific questions, but on that. I thought that it may be interesting to you to see this point of view. So if we start these things, the first thing you know is, let me go back to 1983, that these two papers were the foundation of many things in present plant science. The first paper, you know, the two papers that show that it's possible to transform plants with foreign genes and that it works. And then you can regenerate plants that have these functional genes transformed. I think that we celebrated that 40 years, a couple of years ago. And you see already here, we have one of the questions that are the basis of many of the discussions we have. The first paper that was published in Nature has been published by two public labs. A lab in Ghent, Marfa Montagu was the head of this lab, and Jeff Schell in Max Planck in Cologne. So those were public groups, although, you know, very soon they created a company, Plant Genetic Systems, that afterwards was bought by a big company, but it was public result. Then the second paper in Cell, you see this is in May, this is in April, that created a lot of discussions among themselves about patents, about DI, there was questions raised about that. It was of a company, it was Ciba-Geigy at this moment. People like Patty Del Chilton, she was a very interesting scientist. She was working already in a big multinational company, Swiss origin. Afterwards, that was converted to Novartis. After that, was Syngenta. Now belongs to Chinese. This lab. So those are the starting point of these discussions. And you see from the beginning, we have all these questions. Public science, relations with companies, multinationals, intellectual property discussions from the beginning. And of course, discussions on how to use this new technology. And I asked Will about that because many of you know that in 1990 in Europe, you see, that was 1983. In the States, in 1984, 1996, already they were what they call the coordinated framework for the use of biotechnology in the States. In Europe, it took a bit more of time. It was in 1990. But you see, the first modified plant was planted in the world in 1994. So 4 years before anything was planted, already we had, even in Europe, that things go much slowly. Even that, we had already this thing. And you see, this is the first directive in 1990. And it is, for instance, something like, it is necessary to establish harmonized procedures and criteria for case-by-case evaluation of potential risks. That was the basis of that, of that directive. And following this directive, it was created a committee, a scientific committee of plants that were analyzing. You see, this is 2000. We're analyzing some plants. This one, I was a member of that. You see here, you see me here, and one of the plants notified by Monsanto. For instance, when you start to analyze these plants, you realize this is what's called J21. It gave a lot of discussions between different companies. That was very complicated from point of view of IP, this plant, this specific plant. Also, we discovered when you analyze the molecular analysis that there were not just one insertion, but two. So insertions were complicated. It was never planted, this variety, but it was approved. This was approved. So it was a system put based by scientists. You may recognize some people here, because there's a couple of French people here and other people. So that approved. So the commission requested scientifically to consider whether this is that the placing of the market. This is a herbicide tolerant maize. If there is any risk. So due to that, with this system, a number of plants were approved. Tomato, potato, you probably know many of these things. And finally, maize. And from that, only one, MON 810, from Monsanto, was put into the catalog of seeds. And only this plant is planted in Spain. At this moment, it's still planted. The number of hectares of maize in Spain is being reduced because there's a problem of water. And but I mean, it's still about 30% of the maize produced in Spain still is based on this modification. Why, for instance, in some places near Barcelona, 80% of the maize is this modification because it's a very windy region. If there is the corn borer, it weakens the plant and plants fall down, so farmers lose their plants. So it is, and it also there is a fusarium infection, so it works. In Spain, this modification has worked, and more or less 30% of the maize cultivated in Spain is still this is the only one approved in Europe, the only one. Farmers cannot have the choice, and when they have, they buy freely this modification. So this is the situation that it happens, and you see afterwards it was another directive because there were a lot. In 2000 there was a lot of protests and discussions and conflicts, and it was modified. But you see, This is the system that still is working at this moment to approve. If you look at the journal of EFSA, you will see that every month, a number of new modified plants are approved. They continue to be approved in Europe using this directive, but it cannot be cultivated. It can be imported, but it cannot be cultivated, which is one of the paradoxes of the European system. But for discussions of today, is that here in the center, you see of all the system, in the center, this is the EFSA GMO panel. It's a panel of scientists, 100% scientists, that give their opinion. So in the center of these discussions, there are scientists. And these scientists have different expertise, agronomy, pathology, whatever, environment, and they discuss, they approve or not, or the varieties that are presented. But you see, this here is very clear. This is this division here. Here is what we call risk assessment. This is the role of scientists. But there is risk management. Afterwards, it goes to the European Commission, to member states. They authorize or not, they never agree. So it's impossible to get an agreement in Europe for these things. So the Commission gives the final authorization. So, but you see in the center, so I've been member of this GMO panel in the first parts, and one of the message you always give to my colleagues, go there, be there, because if it's better, it is important that scientists with the best possible information about genetic modifications, about the effects, about environmental, of toxicity, the best opinion, although afterwards politicians do whatever they like. So I think this is one of the questions. We are asked as a scientist to be in places like that. And this decision, at this moment, I think the chairman of this panel is someone from my lab. There are a couple of French people from India in this panel. And then is one of the questions that we are asked, to be in places like that. You see, this is one of the dossiers that I had to review, was in 2001. A dossier of this, maybe 15,000 pages. It cost 10 million euros, I think, or 20 million euros. You see, that was now it's online, but at this point they arrive me with if you know these things, I just keep in my mind. These things, those are very complex. They have molecular characterization, they have toxicity, they have behavior in the land, they have a lot of data that have to be analyzed and people share. People don't read 15,000 pages, of course, at the same time, but I mean, it is, but even if you do that and you say there is nothing against, this was the Pioneer 1507, it was an insecticide resistant maize. Even if you say there is no problem, no specific harm, it may happen that finally, as the system is blocked, the European Commission has to make decisions, they delay it, and when they take decisions, you see, when they take decisions, it may happen that the European Parliament say no, you have to stop allowing this kind of, because it was presented for cultivation. So then after this decision, companies decided to not to present anymore any new plan for cultivation in Europe. And then also it may happen that there is a bomb, that the chairman of the GMO panel receives a bomb. I mean, fortunately it was a stop at the front desk of the EFSA, but you see some people who are absolutely against the use of these plants in Europe. And you see, it was not harm, but I mean, a bomb was there sent to the chairman of the GMO panel. So it's a situation that may happen if you decided to go something like that. So what are the sources of this conflict? Well, there is a perception that some of the users are following sustainable farm practices. Herbicide tolerance is, in Europe, is considered terrible. Herbicide promotes, from the perception of people, herbicide tolerance promotes the use of herbicides, glyphosate, you know, that there is a lot of discussions about glyphosate, about the health effects of glyphosate. There are data from the Institute of Cancer Research of the World Health Organization that says that it may produce cancer at some point. Other reports contradict, especially on the users, but there is a lot of discussion about herbicide tolerance. And the discussions which are at this moment in the European Parliament and European Commission, one of the points is we are not going to allow any plant which is herbicide tolerant. Okay, so this is one of the points that probably will be decided in the next months. So this is still an important discussion. What are the users? I remember when the first plants arrived in the market that some companies were asked, are you sure that to present herbicide-resistant plants is the best idea? Say, big business. Well, maybe big business, but maybe problems afterwards. Okay, so another industrial consolidation. There is a perception of monopoly. It is very clear. You know that consolidation has been happening. That I told you about Syngenta, which is now it's controlled by ChemChina. Well, 2 or 3 big companies, maybe 4, control many of these questions. Because at the same time, this is the patenting. Patenting is one of the big issues at this moment in Europe. And if patenting is a question, patenting allows, the essence of patenting is the creation of monopoly. You have a technology, you are granted a monopoly for 20 years. Okay? So this perception by farmers, by small seed companies, by politicians, say, I mean, they are creating a monopoly. This perception is one of the bases also of the discussions we have at this moment. And well, also, there are some movements that have, so, I mean, they become some industries. I mean, they have their own, I regret to say that, but some movements, they have their payroll to have at the end of the month. So they have to produce some products. And so it is also one of the problems. Okay. So one of the questions that arrive, because science, as you know, science is going fast. What is the definition of gene? This is one of the points that we're discussing at this moment. Because when you introduce a gene of say, tolerance, this is very clear. But then genesis. If you introduce a gene from the same species or very similar root, or side-directed, I mean you introduce something at a specific point, or you do, which is the question now, with the central point now, side-directed mutagenesis, oligonucleotide mutagenesis, protoplast fusion or synchronal variability. Mutagenesis as a whole, this is one of the big discussions we are having at this moment. Mutagenesis is a genetically modified organism. It is a genetically modified organism because you are creating a mutation. But are you going to consider it legally as a GMO? This is very, you know that at this point, the scientific concepts are lost. When you talk about with politicians, you have to be prepared to be asked the most curious of the questions. So, I mean, it is graphic. I mean, what happens if the rootstock is a GMO and the other one is not? Well, you may be asked to say something. I mean, you see, in 2012, this is interesting because you see 2012 is the year that CRISPR-Cas9 system was published. So at the same time, you see there were already opinions, scientific opinion addressing safety of plants developed through the genesis. So it was not CRISPR-Cas9, but it was zinc finger nucleases or other systems of other nucleases that were side-directed. So we were asked, I was in this panel, we said, okay, I mean, if you modify something which is very directed, you modify something with the same gene, you don't have to use the same criteria, not these 15,000 pages, maybe a bit less. Once we gave this opinion, the European Commission closed down this panel because we were saying something that they don't like. So, but you see, these ideas were present, you see, when before the, of course, when the fantastic information we have at this moment, that was in 2000, our group was also part of this result. And you see, you know perfectly that now one of the things that we are faced with is this fantastic wealth of information due to sequence. This has changed completely our work. And also, you see this paper, it was in this year, in 2012, the first demonstration that it works in any system. You can direct this, and then immediately, you know, the same question arises. Still is not settled completely. Still Berkeley and Harvard are discussing about who is the owner of these patents. But immediately this question arises. And this is something that we have to take into account. And so people started to say that it's interesting, this paper, because say precision genome opportunities and regulatory challenges. This is a paper also more or less at the same time. So this is still what we are faced at this moment. So I would like to put this slide because it is a couple of papers that we did in collaboration with a group here in Orsay. Our group was working in Melon, and we collaborated with the group of Abdel Bendaman, just working here 200 meters from here, and we discussed, we discovered that the gene that was providing viral resistance in melon, it is a gene that is related to protein translation, which is something that afterwards has been confirmed in different species. The interesting thing of that is that we discovered a nice paper in Plant Journal. Then people started to look at other alleles using ecotilling. They didn't discover much, but I mean, it was an approach. But then you see, immediately appeared a paper from a group in Israel that say development of broad virus resistance in non-transgenic cucumber using the CRISPR-Cas9 technology. I think that's interesting. I mean, they use our results to create a mutation in the gene, in the EF40, and they discovered that it was possible to create a new viral resistance. Is in cucumber, so it's something which is interesting. I say it's not transgenic. Why it's not transgenic? Because they created this mutation and they make crosses with other cucumber plants, and they get rid of all the modifications that were there. So, I mean, probably they had now some places, some cucumbers with only a mutation, which is providing this resistance and using this kind of results. Probably this, at this moment, that would not be allowed to cultivate this cucumber in Europe. Okay, probably is cultivated in the cell, I don't know. And there are no, you know, in the States, you don't have to notify anything if you use these technologies. Probably they are in use. But it's interesting that very quickly, you see, that was in 2006, that was in 2016. So they use and they use the tools that were possible. And to have cucumber, which is resistant to virus, is a very interesting thing. So just as an example. So we know that for breeding, which is the basis of our farmers, of our farming system, we know conservation. It's important that you know that from the after the Second World War we have, or even before, we have very big places to conserve seeds, to have variability. We had very big efforts to conserve. We have to analyze, because in some places we have thousands of varieties. We don't know exactly what we have. It's an effort that we are doing. We are creating a new variety of mutagenesis, and we are a new variety using genetic modification. Just to remember, you know that mutagenesis has been used in different ways. For instance, radiation. There are a number, you know, like in some crops, we have varieties which have been created irradiating seeds in nuclear reactors. And there are 3,000 varieties of 200 species of plants have been created in this way. And there is no discussion about these mutations that have been created in this way. And also chemical mutations. And we have tilling, for instance, to analyze these mutations. So what are the conditions that we have now? Whether we have methods to transform or regenerate is not as easy as it happens, you know. But those that you have used genome editing, it is something that can be used. In some species, still is not possible to make this. So we have to have systems to transform and generate plants, and maybe not this in some species or some varieties. We have to be able to control of targets. We have to remember these things, and to have especially candidates that are interest for breeders and farmers. If you know that there are some databases that people are compiling what is being done at this moment, different things, resistance, quality of the, for instance, traits related to drought resistance, for instance, are being used. So there are a number of questions, especially you say from USH, you say that rice is the most important crop being used in tomato. So these things are hard. But you know that in Europe, technology is going very fast. The results are being published. But you see the European Court of Justice in 2018 decided that this is a GMO. If you use something which is using the CRISPR-Cas9 or similar things to create a mutation, you have to consider it as a genetically modified in the terms of the 2001 directive. Okay? You may be surprised as a scientist of this result, but it seems that from the point of view of the juridic, the way that laws have been created and discussed, this is perfect. It's a perfect judgment. It's what the court, it is formed by some of the best lawyers in Europe, decided that this is the question, and this is the so there are questions. I think that has been a number of reports. It is interesting also, I want to share with you that some of these reports, maybe if you, as I did myself, produce reports, ask with politicians in Brussels, you may find yourself for instance, a report produced by the European Parliament, the group of Greens, they say, P Puigdomènech, you see, is involved in many lobbies, say something like, Puigdomènech has authored numerous joint publications with industry. It is untrue, it's false, it's a lie. I never publish these people. But you see, some people come with me, make a thesis with me. Afterwards, they go to companies to work with companies, which I am very proud of. And then you see, in the report say that I am publishing with industry, which is completely lie. But it's paid by public money, European. You see, and you may find yourself in something like that. You see, in the States, there you go. There are a number of opinions. I took part on that. I think that a report which is interesting, if you are interested, this is Reimagine Europe, is a think tank in Europe, produced a couple of reports, especially recommend to all these things. Existing narratives shaping the debate. What people produce, what kind of arguments people produce in favor or against. It is an interesting report. Policy options, well, I hope, it has been a number of. Let me just, you see which we are at this moment. At this moment, we have a trial going on. The Council has approved modification of the directive. The Parliament has approved another one. The Commission another one, and they have to agree. Is what we are at this moment. Yes, gently, just to finish one things. Patents. This is one of the points. We produce, if you are interested, we have produced last year an opinion in ALEA, the Association of European Academies, the statement of the measures to ease the impact of IP system for crop development. I invite you, this is open, you can go there. There are a lot of discussions about how to minimize. Farmers are worried, small companies are worried, so it was some question. So let me just make my last point. When we work in science, at some points we say that we have the global situation. You see, people who are problems for having problem accessing to food. It has been decreasing, but now it's increasing once more, especially because of there are wars, conflicts all over the world. But there is a real problem there. We are not reaching the goal of ending hunger, so we are using this argument. Of course, we are using. But the question is, and the population is increasing, and we know that food-related emissions is more or less 30% of the total emissions. So they have a problem for climate change there. And you see, farmers may block INRA, maybe saying to INRA, what are you doing, scientists in INRA? You are saying, for instance, that you have to reduce pesticide use, but what are the solutions for us? Are we, as a scientist, giving solutions to these people? This is the questions that we have to, because they are complaining. So we have to see. And actually, I've been part of some ethics committees. Because this is, of course, an ethics question, what we are doing with our science. One of the, for instance, here is an interesting statement on the Danish Council of Ethics. They say something that it is ethically relevant to reject GMO varieties if they can help to alleviate or solve significant problems, and there are no good arguments for rejecting. It's the result of an ethics reflection from Denmark. But it is. INRA, Sirati Fremer has a committee of ethics. I have been a member of this committee for 12 years. and we produce a number of reports. I recommend you, if you know, to go and to see, because we produce a report on new techniques. And also, in Europe, there is the European Group of Ethics of Science and Technologies. I'm the member also of this committee for 13 years. And we produced this report in 2008. I think it's important that some scientists take part in these committees. And there are philosophers, there are jurists. They have very interesting ideas. They don't know normally nothing about science, and they talk about science. Right? So it's important that some scientists are there to try to inform these people, right? To give arguments. This report is it's quite a complex one. I think it's still goes on. I think it's have interesting, good ideas. I think that the ethical approach who considers that the goals of new technologies have to try to solve questions of food security, food safety, and sustainability. I think that still these are the goals that, from this point of view, are important ones. And lately also this committee of INRA has produced this report, especially what are the rights and what you have scientists have to behave when they are convinced that there are important problems. Here it is about environmental change, but food production is also an important problem. And he says that what the scientists have to do is integrity, deontology, and humility. It's what is produced, how scientists have to behave when you talk to society about these questions. Okay? Thank you very much.